All right, got it all going. And I replaced the batteries in the microphone. I apologize. I realized after the class got done, I'm like, my microphone was off. So I'm like, I didn't know when it stopped. And then I looked and I was like 5-6 minutes into the recording and my microphone died. I wish it would give me warnings or beeps or something, and it does none of that. So today, it's a one PowerPoint only unit, so it's like a little mini unit. There's no big exam on it. Instead, there's a worksheet that I'm going to hand out today. It covers Chapter 9, and I don't lecture on Chapter 10. There's a couple questions about Chapter 10 on the worksheet, but Chapter 9 is all about genetics. So DNA, that transcription, translation, which hopefully should be some review from previous classes like AMP. Again, we'll look at some differences in how bacteria do transcription translation differently. As we start talking and getting into bacteria and viruses, how some viruses can do this a little wonky too. So some of this I'm hoping will be a little bit of review, but whole PowerPoint on genetics. Whatever I don't get through today in class, which it'll probably be pretty close that I should be able to get through it all, I'll record it. It probably will be 5-10 minutes left, and I'll put online is the remainder part of the genetics lecture. So the genetics lecture, if my thing would work, nothing can all work today. We're going to go over what are some basic terminology in genetics, looking at what is the structure of DNA, how does DNA replicate, going through what is transcription, what is translation, talk about genetic mutations that can happen, and then also talk about genetic transfer. Because again, when bacteria specific talk to each other and exchange DNA, it's good for them, especially if they're exchanging information on how to be resistant to antibiotics, but it's bad for us as they share that information on how to be resistant to antibiotics. So some basics on just what is it. So what is genetics? It's the study of inheritance, and whether you want to shorten wordage on this to however you want. Inheritance and heritable traits that are passed on all based on an organism's genes. So if you guys have a gene that expresses your hair color, your eye color, that's a gene that can then get passed on. That's what genetics is. We're looking at traits based on your DNA, based on your genes, that can then be passed on. They're inheritable. And it's the same thing. If bacteria have a particular gene that codes for, say, having a capsule or having flagella, granted, they don't pass it on to baby bacteria, but as they grow and divide, that information gets still copied and kind of a copy paste to every new bacteria that's made. When we talk about organisms and their genome, we're looking at what is the entire genetic makeup of an organism. We want to know the DNA sequence of every gene. We want to know every gene and everything those genes code for. We've finally figured out, just I'd say in the last 10 years, the human genome. For a long time, that took 20, 30 years. They had the big old genome project. We want to know human genes and what do they all code for, and we want to know their DNA sequences. We've done this for a few other animals and insects and things like that as well. Otherwise, we just don't spend the time. And that includes every gene that it has, every DNA sequence. So that's your nucleotide sequence. And we've coded it for all the humans. We've also coded it and figured it out for a lot of different bacteria. Because again, they talk. We want to know what's going on, especially if they cause us issues. Now, what do you know about DNA? So some basics of DNA, I just put pictures up here. Again, review things as just if you forgot. Because you should know some basics of DNA from previous classes you've had, probably from middle school all the way to A&P. Basics of DNA. One, there are 4 DNA bases: adenine, thymine, the guanine and cytosine. The uracil is RNA. DNA, when we've got these bases, your adenine, thymine, guanine, and cytosine, they pair up that way. A's always pair up with T. In DNA, G always pairs up with C. They do it generally just based on their size. We always have one big structure pairing with a small structure. So like one big double ring structure pairing with one small structure. One big double ring structure paired with one small structure. It keeps the width of DNA the same if you always have one big with one small. They are held together when you've got the G's and C's or the A's and T's. They're held together with a hydrogen bond. Why that matters, hydrogen bonds are weak bonds, which means DNA can get yanked in half quite easily or unzipped, as you'll see it. When DNA needs to replicate, we have to yank the two strands apart. So luckily, they're held together with a weak bond. In the structure of those adenines, thymines, guanines, and cytosines, that's that nucleotide base. It's a nitrogen base. But on the outside of that nucleotide structure, we always have some type of sugar attached to some type of phosphate. So there's always a sugar, phosphate on the outside. Does anyone remember what sugar we find in DNA? Because it's part of what DNA's name stands for. First, what does DNA stand for? Does anyone remember any of it? You remember some of it? No one remembers any part of what DNA stands for? Anything? You're just like, I don't know, some letters. Deoxyribonucleic acid. You're like, oh yeah. Nucleic acid, because that's what, nucleic acid, up there. That's what these are the nucleic acids. The deoxy part is based on the name of the sugar. The sugar that we find on the outside part, they call it this just like outside chain. It's purple. That sugar we find on the outside is called deoxyribose for deoxyribonucleic acid. A couple other things about DNA. So I was like, it's a double strand. So you've got two outside strands that attach together. It does form a twist, a helical shape. So it's the old school telephone cord. It's got that helical twist to it. But another thing that we'll talk about today that can cause issues when the DNA has to replicate is that the two strands run opposite of each other. They go in different directions. It's called anti-parallel. So even if you notice what the sugar and the phosphate look like, on the other side they're upside down because they run opposite directions of each other. When we draw out DNA, the two strands, based on whatever is the very last sugar structure and how it's oriented, one side is an organic chemistry naming. It's called the 5', that's what the little dash means. And the other side is 3', which means if they're anti-parallel, the other strand has a 5' on the other end, and then the 3' on the other end, opposite of each other. Again, it's based on the structure of the sugars. So on this is the 5', which means the other end is the 3. And it's because they're anti-parallel, if this is the 3' on one side, it's the 5' on the other versus the 3. So here's the 3'. If we follow it all the way to the end is 5, and then at the opposite. That can become an issue, as you'll see today, when the DNA is replicating and making new DNA. It's the fact that the two strands run opposite of each other. Now, the structure of a prokaryotic genome, and in our class, we really just care about the genomes of bacteria. I don't care about anything that's an archaic organism. But a prokaryotic genome, so this is the genetic material in a bacteria. It includes all of their DNA and any associated proteins that may have to hold it all together, and any possible RNA. Their chromosome is haploid, which means they just have one copy. So where our chromosomes are paired, we have 23 pairs of chromosomes. Bacteria just have one copy. Nothing's paired. Bacteria's chromosome is generally circular. Now, the circular can be a big enough circle that it starts to twist around, but it's still one continuous circle. And because bacteria don't have a nucleus, that genetic material just hangs out in the nucleoid region, which just means nucleus-ish. There's no actual envelope structure. Bacteria can have plasmids, something we can't have, we don't have. But bacteria have plasmids. They're small molecules of DNA that are separate from their original chromosome. So bacteria, not all bacteria have this, but bacteria, they've got to have their chromosome. This codes for everything that they need in life, for growth, survival, everything. But bacteria can, a few other organisms can have them too. Bacteria can have these little snippets of bonus DNA. They're not needed for survival, so they're not essential for everything else, growth, reproduction, that's all part of their normal chromosome, but it's just extra DNA. And if that extra DNA is the correct sequence, it could code for certain genes. And a gene codes for some feature. And so it can have some survival advantages. So they need this to survive. That's your basic survival. But maybe this gives them a little bit of an advantage, because maybe there's a gene found on this little bonus DNA that codes for how to be resistant to an antibiotic. In the video, The Hunting the Nightmare Bacteria, David had an antibiotic resistance bacteria. Do you guys remember what the gene was called? It was some letters. It was NDM1. And NDM1 is a gene, a little snippet of DNA found on a plasmid that made whatever bacteria that had that gene on it able to resist almost every antibiotic that we have. And the problem is this can't leave the cell. This is like it needs it to survive. But these little snippets of DNA, the plasmids, they can replicate and they can leave the cell and go infect other bacteria, which was what was happening in David's leg. The bacteria were all chatting, and this bacteria was sharing that information on how to be resistant to every antibiotic with other bacteria. Now, eukaryotic organisms like us, as well as fungi and anything in that whole eukaryotic domain, they usually have more than one chromosome per cell. Chromosomes are diploid, which means they're paired. Like we have 23 pairs of chromosomes. The chromosomes are generally linear, so if I drew your 23 chromosomes, I'm usually going to draw them as X's, or if nothing else, just the line if they haven't duplicated yet. I'm not drawing circles like I would for a bacteria. And we find all that information in the nucleus. Now, some fungi and some protozoa, which are still eukaryotic organisms, can have plasmids too. So plasmids are not a bacteria-only feature. Some organisms can have plasmids too. We can't, but some fungi and other protozoans can. Now, DNA replication. Anytime any cell needs to make more cells, they have to duplicate everything. So all the organelles before it makes new one, but it also includes any new DNA. So anytime a new cell is formed, we need double the DNA. To make DNA, some of the things we need the building blocks, the monomers, and we need energy to put it all together. So a monomer just means single things, and then we have to put it all together, because mono means one. So I always like to think it's like the bricks. You can't make a building without first putting all the single individual bricks together. We can't make DNA without putting all the individual pieces together. So we need all the pieces, and then we need the energy to do it. Now, what's nice is that we do have some structures that have both of those things, that have the brick, but they got the energy with it as well. They're called triphosphate deoxyribonucleotides. It's a mouthful. It's still a nucleotide for DNA. It still has the base. This one is using guanine as an example. It still has the sugar deoxyribose, and it still has a phosphate group. That's what makes our nucleotides. But because it has extra phosphate groups, we have extra energy. It's the same basic idea of ATP. ATP is adenosine triphosphate because it has 3 phosphate groups, and we can release energy if we break bonds. And then ATP becomes ADP, or adenosine diphosphate. Well, if we have a triphosphate deoxyribonucleotide, you have the whole base, but because you got extra phosphate groups, we have the energy too. Oh, we need a little energy to put a couple bricks together. Let's just break one of these bonds. You break a bond, you release energy. So we've got the building blocks and we have the energy source to put it all together. When we make DNA, new DNA, when it replicates, it's also done in what's known as a semi-conservative model. So if we had one double-stranded DNA, but we have to duplicate it, so now we have 2 double strands of DNA. What the semi-conservative part means is in our new strands, our 2 double strands of DNA, one of the strands is always from the original DNA. That's semi-conservative. Is we know that in the new DNA that's made, one of the 2 strands in our double-stranded DNA came from the original strand. It served as the template to make the other side. And for a long time, we didn't know that. We didn't know if, like, well, we had this double-stranded DNA, we yanked it apart, we made 2 red strands, and we put the red strands together. And so we had an original and a fully new. We eventually figured out there's always one new red strand attached to an original template strand. Now, to make new DNA, several things have to happen. So there's your double-stranded DNA. Now, DNA wants to twist. It wants to form that helical shape. And when we have to make new DNA, take that original strand and make it as a template, we do add some stabilizing proteins. So if you're wondering, what the heck does a stabilizing protein do? It prevents the twisting while undergoing the replication. Because you have 2 strands that are held together, but we have to yank them apart to make them, because we have to yank them apart so that each one becomes a template that we are then copying to make the red strand. There has to be something that yanks the 2 strands apart. It literally is called unzipping, and that's an enzyme called DNA helicase, which I think I've got. There you go. There's so many big memes. If I can find a meme that's a Big Bang Theory meme, I throw it on there. So the helicase enzyme is unzipping or pulling the 2 strands apart. So now you've got your 2 template strands. Now, there's other enzymes involved as well. There's one called DNA polymerase. The job of DNA polymerase is to bring in the correct bases. So if your original DNA strand had an A, what base would get brought in? What pairs with an A? In DNA, though? A T. If this strand over here, I don't know, maybe this base is a C, what base would get pulled in? A G. And I'm like, so that DNA polymerase is the one that's bringing bases in. But it has an issue. It's because when it is, because as this is unzipping to the left, and we're trying to work that way, it can only bring in bases, the A's, the T's, the C's, the G's, whatever it needs to bring in, it can only bring in bases and add it to a new 3' end. This becomes an issue. It can only add it to a 3' end. So this is showing the 2 different strands and how it manages that. So again, it's unzipping to the left. And on the very top strand, that dark blue, that's your original template. And on the end is the 3. So that means my new strand has to be the opposite. So my new strand starts with a 5. And then I'm adding more bases going to the left. I'm unzipping that way, so I'm adding more bases. That DNA polymerase can only add onto the 3' side of this new strand, which is just fine. Because if it starts with the 5, the other end is a 3, and that's where it's adding the bases. So that one doesn't have any issues. But the other strand has an issue. Because if my original strand starts with a 5, I can only add on to the 3' side of the new strand. But I'm unzipping going to the left to make my new strand. I'm bringing in bases, but this is the 5' side, and DNA polymerase can't add on to a 5' side. It can only add on to a 3' side. So it has to do the other strand in segments, and I'm going to show a little video of this. It has to add it in segments. So it uses other enzymes. I don't care that you know other enzymes, but it will start, we'll just say right here, it will lay down almost like a little start sequence, and then that RNA polymerase goes backward, adding onto a 3' side. Then it jumps ahead again, adds almost a little start segment, and then it goes backwards so that it's still adding onto a 3' side. So it has to keep jumping ahead and going backwards, and jumping ahead and going backwards, because it can only add onto a 3' side. Then other enzymes come in and clean it all up to make it one continuous new segment. So that's why this is called the leading strand, because that's no issues whatsoever. It doesn't have to go backwards. And then there's the lagging strand, because it takes a little bit longer because it has to keep doing it in little tiny chunks and going backwards. The little chunks that it makes as it goes backwards are called Okazaki fragments. Now I was making sure my audio works, but we'll see if it actually works while it's doing stuff. Ignore the background little music stuff.
When DNA replicates, its strands are separated.
Now my little note, all my little pictures, it was unzipping going to the left. This video has it unzipping going to the right. It still does the same thing, it just is backwards.
Separated by the enzyme helicase.
That's the thing unzipping.
Single-stranded DNA binding proteins keep the strands from reannealing.
Stabilizing protein.
One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA. The process repeats again and again. DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
It's like the background music's annoying. But it's showing again, the leading strand has no issues because that DNA polymerase can just always add to the 3' of a new strand. It's that lagging that becomes the issue. Now, your genes they code for certain traits. Again, you have a gene that codes for your eye color, you have a gene that codes for your hair color, you got a gene that codes for just about everything about you. Now, a genotype what are your genes and what do they code for? A phenotype is like an outward physical feature. So you have genes that code for things. So your genotype is you got a gene that codes for eye color. Eye color would be the phenotype. The gene is the actual genetic material that codes for that phenotype. But I do specifically put your genes don't always show themselves as a phenotype. So we have a gene that can code for flagella. So you've got a genotype, you've got a gene that codes for a specific feature like flagella. But the reason why I put the not equal is even though you've got lots and lots of genes, it doesn't mean they're always expressed or turned on. You guys have lots of genes for lots of different things. You guys might have genes for black hair, but you don't have black hair. Maybe you've got a gene that codes for red hair, but you don't have red hair. I'm like, you've got lots and lots of genes in your DNA. And I know there's more than one gene that codes for different eye colors and hair colors, but certain genes are turned on and certain genes are turned off, which also gives us a lot of variety with things. So genes do code for phenotypes, but they're not always expressed. Now, how do we get the information that's on the DNA to actually do what it possibly can do? How do we take the information in a gene and cause it to happen? So we're specifically going to be talking about the genes and the proteins that those genes can code for. Enzymes are proteins. So, because I think it's part of the worksheet that I'm going to hand out. We talk about, I think it's lactose catabolism. What does lactose catabolism mean? Going back to different words. What does catabolism mean? Breaking down, because cats break things. So lactose catabolism means? Breaking down lactose. There is a gene that codes for that because it's the gene, it's the information that codes for making a specific enzyme. What enzyme breaks down lactose? Any guess? It's an enzyme. What do enzymes like to end in? Ase. Lactase is an enzyme. Enzymes are proteins. Lactase is the enzyme that breaks down lactose. So it's an enzyme, it's a protein. That means there's a gene. If you guys can drink milk without having any issues, your lactase gene is doing great. If you're like, no, I can't drink anything that has dairy, anything that has lactose, you're lactose intolerant, it just means your gene is not turned on. And unfortunately with aging, this particular gene starts to shut down. So if you could have drank all the milk as a kid and now you're like, I can't drink any milk, your gene is slowly turning off. Now, we're just going to use our lactase and making this gene as our example today. So how do we get the information that's in the DNA? Because the DNA contains everything, all the information, all the DNA, all the genes that you could possibly have. How do we make lactase? How do we find that gene and make that particular protein, make that particular enzyme? Because the DNA in a eukaryotic organism has to stay in the nucleus. It cannot live. I like to always compare it to like a super, super important book, like the Book of Life that will tell you everything about life. And there's only one copy of that book, and it's housed in the library. Are they going to let you take out that book from the library? No. It's like one copy that explains all of life. That'd be great if they had it. But that's what DNA, it's like the one copy of your information that explains everything. But I don't need everything in a particular book to know how to do this. I just need maybe paragraph 3 on page 100. I'm like, I just need a little bit of information. That's what a gene is. It's just a little bit of information that tells you one thing. I don't need the whole book of life. I don't need all the DNA. I just need a gene. But because the book can't leave the library, just like DNA can't leave the nucleus, how do we get that information from that page? You make a copy of it. Can you make a copy of page 100 at the library? Sure. You could take it to the photocopy machine and make a copy of that page. That page then can leave the library. The book can't. That's what's happening in transcription, is you are making a copy of the DNA, but specifically just the gene you want. So I only want a copy of the information on that paragraph. I'm just making a copy of that one small little gene, that one small little piece of information on what I need to know. And for this, our need to know is how to make lactase. That's transcription. Transcribe means to make a copy. If you're a transcriptionist, you like copy everything you hear. Transcribe means to make a copy. But just because you have that copy of page 100, and you now have that information that you can walk out of the library with that page, you haven't actually made the lactase enzyme. So you have to take that information that's on that page, and it's called translation because you translate it into a different language. You translate it into the language of however it's written on that page, into the language of proteins. So I take the language of words, because you read it in the library, into the language of amino acids and proteins. That's translation. And we're going to go through this. It's called the central dogma, of genetics. Erase that. Is that we can take the information that's on DNA and we can transcribe. I can't talk and write. I don't want to just write transcribe. Transcription. And make messenger RNA. Now, it's not DNA. Again, I'm not actually ripping out page 100. I'm just making a copy. It has all the information I need, but it's slightly different because that's not the original page. I can take that information, that little snippet, that little copy of information, and then I can translate it into a different language. The language of proteins. A polypeptide just means a lot of amino acids. Amino acids make proteins. So we're going to break this down. This is what a lot of your worksheet's going to be, is taking, if I give you a strand of DNA, can you transcribe it, then translate it? So up here, I've got my double-stranded DNA. And in transcription, when I make a copy, I'm just copying one of the strands. I'm not copying both strands. I just need one strand that has all the information I need. So it's going to transcribe the bottom strand. Now, to make RNA, if the original DNA strand has a T, it will bring in an A, because A pairs with T. If it had an A, instead of bringing in a T, RNA doesn't have T's. It has uracil instead. So you won't see any T's whatsoever in RNA. Instead, they're replaced with U's. They look similar enough. They know where to pair up. If it had a C, it will bring in a G. If it had a G, it will bring in a C, and so on. It's now made a copy. And again, it's not the exact same because it's got U's instead of T's, but it's a copy of the information. It's called messenger RNA because it's the message I need, and the message can leave the nucleus. Because you can walk out with that sheet of paper. Then it's going to make proteins. Ribosome's job is to make proteins. So of course, the ribosomes are going to be involved in this. The ribosomes are going to read this information. And they're going to translate it into a language of not DNA, but it's going to change it into a language of amino acids. And it reads it 3 letters at a time. These 3 letters code for this amino acid. These 3 letters code for that amino acid. So it knows which amino acid should it get brought in. It's just reading it in a different language. Now, more slides to keep going over this. So participants in the transcription. So we have to have the original DNA that's going to be copied. We, of course, have to have more enzymes because they do the job of everything. So the enzyme job that brings in the correct RNA bases, the correct RNA nucleotides, is RNA polymerase. DNA polymerase brings in DNA nucleotides. RNA polymerase brings in RNA nucleotides. So RNA polymerase is the one that's bringing in the nucleotides. RNA polymerase, it's the one that's reading this as a T, and it brings in the A. It reads the A, it brings in a U, it reads the C, it brings in a G. Now, there are a few other RNA molecules out there. Messenger RNA is the only one I want to focus on today. There's a couple other RNA molecules as well. They're more structural, but we'll talk about tRNA too. Now, why? I want to find this video because it's going to show you transcription in real time. I wonder if the other video clip worked. Probably not. Sorry, I'm going to YouTube. I should have checked that one when I was checking audio. Can't spell today.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell.
Oh, I feel like this is just the translation step. Hold on. I don't want that one. They used to have as one video, not two.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a...
Now, first I'm going to sit because I'm going to pause this. Pink structure, what is it? Based on its appearance. It's the DNA. So again, in transcription, we're going to take the original double-stranded DNA and make messenger RNA. Now, messenger RNA, if I go back to my PowerPoint, is only single-stranded. I'm only making a copy of one side of the original DNA. But here we're having some of those proteins, those enzymes that are going to be unzipping helicase and reading it.
At a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase.
The thing that's bringing in AU, C's, and G's.
Into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top. From the long messenger RNA chain
So again, it's unzipping the double-stranded DNA. It's reading it one letter at a time, bringing in the correct bases. So if the DNA had an A, it would bring in a U. If it had a C, it would bring in a G, and it's making that messenger RNA. It's not copying the whole DNA, just one small gene, one small segment. Now, once we have that segment, once we have that message that leaves, so here's your messenger RNA, one tiny little segment left the nucleus or the nucleoid region, we then have to translate it into the language of proteins. Since ribosome makes proteins, they're going to be involved in this process. So the ribosome is going to read that information on the message, and it's going to bring in amino acids. It's the job of a molecule called transfer RNA, which sometimes you'll see it as a little t next to RNA, the transfer RNA that actually brings in the amino acids. So transfer RNA can see AUG, and it will bring in methionine. Transfer RNA can see CGG, and it will bring in arginine. It can see the GUA and bring in tyrosine. So it's called transfer RNA for a reason. It's transferring in the correct amino acid with the correct RNA sequence. But it always reads it 3 letters at a time. Every 3 letters is called a codon. It's just another term, but if you see it. This is the structure of that transfer RNA, just done in different, kind of more of a 3D type image. How the transfer RNA knows which amino acid to bring in is on the bottom structure of that transfer molecule, there are 3 letters that matches up with the codon. And although it matches with the codon, how it matches is called the anticodon. So if the original strand had UGG, it knows like, oh, UGG, well, ACC would match with that. So this is the correct transfer RNA to bring in the correct amino acid. So it doesn't just truly read it because it's not that evolved, but it has the 3 letters to match up with it. Now, there are 20 different amino acids, but there are 64 different ways to mix AU, Cs, and Gs around into 3-letter combinations. So 64 different possibilities of mixing AU, Cs, and Gs into 3-letter combinations, but only 20 different amino acids, which means there are some repeats. For example, all 4 of these codons, these 3-letter sequences, they're all different sequences, but they all code for the same amino acid. Again, there's 64 different combinations, but only 20 different amino acids. And the transfer RNA can read those different codons. Now, there are some bacteria that have a couple other different amino acids, but I'm not going to focus on those because they do have a couple more than just the 20. But if you notice there's some colorful regions in there, is that the ribosome has to know where to start reading the message on messenger RNA. What ribosome is always looking for is the 3-letter sequence of AUG. Anytime it sees AUG, it's like, that's where I need to start. That's the first letter of the first paragraph that I need to start reading. And so it's called the start codon, and it brings in the amino acid methionine. If we can just shorten it, you can shorten it as well. And then it will continue reading 3 letters at a time, bringing in the correct amino acids until it encounters one of these 3-letter sequences where there is no amino acid, except in some bacteria have a bonus amino acid we don't have. But it encounters one of those 3-letter sequences that there is no amino acid to bring in. As soon as it encounters one of those, it's like, oh, I'm done. I can't bring anything in. This is the last period in your sentence, in your paragraph. And then it's done. Now, reading this table, because you're going to have to read this table for your homework today. If you're given a 3-letter sequence, instead of being like, all right, my 3-letter sequence is AAU, instead of being like, where is it? There's an organization to the table. On the far left-hand side are the first nucleotides. So if you're looking for AUU, your first letter in your codon is an A. So on the far left-hand side are your 4 options. Our first letter is an A. So we know we're in this row. Everything in this row all has the first letter as A. So right there is going to narrow it down. Your second letter is a U. Across the top are the 4 options. As what is your second letter? Well, our second letter is U. So we know we're in the 3rd row, but now we also know we're in the first column, which narrows it down to we're in this box. The 3rd letter options are shown over here. And our 3rd letter is a U. So we know we're AUU. So we're at the very, very top of the box. So that's how you can read the table. So try to find the first letter, second letter, and usually once you're in the box, it's pretty easy. Ribosomes, just looking, their job is to read the messenger RNA. Ribosome structure, whether it's prokaryotic or eukaryotic, they both do the same job. Structurally, they're a little size different. But ribosomes are actually 2 molecules that come together. If you never realized it, ribosomes are actually a 2-part molecule. If it's a prokaryotic ribosome, they're a little bit smaller, but they come together. In a eukaryotic ribosome, they're a little bit bigger, but it's 2 molecules that come together. And they come together around that messenger RNA sequence. This is showing an example. There's your ribosome. So here's your messenger RNA. That's going to get read and translated. There's the ribosome. The 2 parts are going to come together as soon as it sees. It's like, oh, there's my first word. The two pieces are going to come together. Now the ribosome is going to pull that message through, reading it 3 letters at a time. So it's going to bring in methionine. It's always the first one. Then it'll read the next 3 letters, your GGG. And the transfer RNA is like, oh, I know, I recognize GGG because it's got the opposite. And it brings in the correct amino acid. Then it gets pulled through farther, and it will read the next 3 letters, GCC, and it recognizes it, and it brings in alanine. And then it will keep pulling it through, reading it 3 letters at a time, bringing in the correct amino acid. Now, when you get a chain of amino acids together, as these little purple circles are shown up here, when you get a chain of amino acid, that's called a polypeptide. Poly means many. Peptide is referring to the bond that's holding amino acids together. It's not technically a protein yet. All you've got is a long chain. It's like I haven't made my sweater yet. All I have is a string of yarn. Once you shape it into the correct shape, then it's the actual sweater. It's the actual protein. But first you need to make the yarn before you can actually make and fold it correctly. It will continue to keep pulling that message through until it encounters one of those 3 stop codons, and then it's done. This is just showing another example that 2-part ribosome kind of grips down on your AUG start codon, and it brings it through. Ribosomes are small, and there's kind of 3 spots in there. I don't care that you know them too much, but if you see it, and you're like, A is the first site because it's getting pulled in from the right-hand side towards the left. A always means just approaching. It's like the first site. It's just starting to read it. P is for peptide. It's making that peptide bond to join the correct amino acids together. E is for exit. It's now leaving because it's bringing more message in. Now, we have to make a lot of proteins. Bacteria have to make a lot of proteins. Every enzyme is a protein. So proteins are enzymes, proteins are structural materials, and proteins are everywhere. To speed it up, because we have to make so many proteins, lots of different ribosomes can read the same message. It's kind of like, well, if you made a copy of page 100, you can have 100 people all like looking over everyone's shoulders to read that page 100. You can have multiple ribosomes all reading that same message at the same time. So this is an actual picture, electron microscope, an actual picture of lots of different ribosomes all attaching to that message. They're all in order, reading that message, all making the same protein. It speeds it up a little bit. I'll go back to the video. Might have to, I want this one, but I'm going to scoot. This is the original one I wanted, but I'm scooching. Because I don't know who this guy is talking.
A copy is complete.
Okay.
It snakes away from the nucleus and into the outer part
So
of
it's
the
just
cell.
made that message, the messenger RNA, long strand,
Then,
left the
in
nucleus.
a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome.
2 molecules.
It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process.
There's kind
Inside
of the 3
the
sites in
ribosome,
there.
the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
What is insane? We turn out 2 billion molecules of hemoglobin every second. So again, you have to think 2 billion molecules of hemoglobin every second. Lots of ribosomes reading it all very quickly. Yes. Now, we're going to practice one because this is what you guys are going to be doing on part of your worksheet. But just comparing transcription and translation, prokaryote and eukaryote. The basics are the same. Few differences, though. In a prokaryotic organism, their chromosome is one big circular chromosome. They still have to pull the 2 strands apart, so you've got your blue and your pink. And it's still going to do transcription, which is not in the nucleus, because there is no nucleus. And it's going to make messenger RNA. So that's the green strand. Then it's still going to have ribosomes, which are a little bit smaller in a prokaryotic cell. And the ribosomes, which is showing 3 different ribosomes, all reading the same message at the same time, still making the protein. Nascent just means building. So these are like building proteins. They're still growing. The only main difference in difference in structure of the chromosome, in a eukaryotic organism, chromosome is linear. It's not a continuous circle. But the messenger RNA, the making the copy, happens in the nucleus because eukaryotic organisms have a nucleus. That would be comparative to the library. If you're going to make a copy of page 10 or 100 or whatever, that copying will take place in the library. That message then can leave the nucleus, and you're still going to have ribosomes reading it. They're just a little bit bigger structure-wise. Now, I give you a DNA sequence because I'm going to do this on your worksheet today. Can you tell me the RNA sequence? And then, can you tell me the amino acid sequence? And you'd have to squint probably. So, before we get to that, if there is a T on DNA, what letter gets brought in for RNA? A. What letter gets brought in if there's an A? A U, because again, there are no T's in RNA. There is a C, a G, should be AUG. First letter is an A. C. A. Yay. That's transcription. Ooh, and it's going upwards. Amino acids. You're going to have to use this table. AUG, find it. MET. You guys can shorten to the 3-letter codes that are on there, please, for your worksheet. Try to shorten it. Find UAC, but don't say it yet. Is that MET? Yep. Find UAC, because I don't know what it is. Just don't say it yet until everyone finds it. And again, first letter is a U, so you know you're going to be in the top row. Second letter is an A. You're now going to be in that 3rd column. What amino acid does UAC code for? TYR. Find GGU. What does GGU code for? The glycine, down on the bottom here. What about UUU? Yeah, PHE, the very, very top left one. And what about UAA? Stop. Now, you're going to have to do that on your worksheet today. My only note, because I usually get a student that asks me this question. Yeah, it's an actual DNA sequence code of the enzyme that codes for lactase. But the actual DNA sequence code that codes for lactase is 22,000 letters long, and I didn't think you guys wanted to do that much. So it will not end in a stop codon. Because it's like, well, there should be a stop at the end. Yes, 22,000 bases later, I just give you the first snippet of the sequence. So if you're like, why is there no stop? And if you have a stop in there, in the original sequence, probably shouldn't. Because it's an actual part of the sequence. But there's an issue. There's a reason why we haven't gotten to that yet. Is mutations can happen in DNA sequences. So a mutation just means any change in a DNA sequence. Any change. Now, mutations themselves are rare, and generally change is bad when it comes to DNA sequences. And usually mutations to DNA kills the cell, because the cell can't do what it's supposed to do, because you've changed the DNA sequence. Rarely, but it can still happen, that a change in DNA codes for an advantage. Sometimes it does. Sometimes. Very rare. Usually change is bad. Very rare, like 1 in a million, maybe 1 in a billion times that you change a DNA sequence and it codes for something new that's actually an advantage. This is when we get into, if it codes for an advantage, then you get natural selection. If it's something that can then get passed on. Like that change in DNA stays and it can get passed on. Natural selection. Examples, if this bunny is not supposed to have stripes, but depending where this bunny lives, maybe having stripes is an advantage. It can blend in with grasses better. And it survives over all of its, friend bunnies. They all die, they all get eaten, but it survives and passes on its genes. Natural selection. Rare that mutations code for good things. Now, a gross mutation means gross means large, a large change in DNA. So there's a lot of change in the DNA. A point mutation means 1 letter changes, 1 base. And if you're like, well, that's not so bad, depending what base it is, it could still be deadly. Now, depending on what base is changed and what's the outcome of that 1 change, there are different types of point mutations. And on your worksheet, you're going to have to know these two because I ask about them. So I give you, at the very top, this is normal DNA, transcribed normal messenger RNA, translated normal amino acid that will get brought in. That's what it's supposed to do. But we're going to change 1 base and see what happens. So on here, instead at the very top, the 3rd letter was an A on your DNA, but I'm going to make it a G. It's a mutation. It's a point mutation. I changed 1 letter. So instead of a U getting brought in, a C gets brought in to pair up with that mutated 1 base. But the ribosome, when it reads it, UUU codes for phenylalanine. UUC codes for phenylalanine. Does your amino acid sequence change? No. It's called a silent mutation. You can still make the correct protein. Outwardly, you shouldn't have any change whatsoever. You're going to survive, no issues, but your DNA is different than what it was supposed to be. So it's called a silent mutation because you don't actually see the mutation occurred. If you're going to have a mutation, that's the best one. Missense mutation. So again, my original DNA up there, but I changed this letter. So it was a G, now I made it a C. Still just 1 letter changed. Everything else stayed the same. I brought in phenylalanine, I brought in tyrosine, but when it came to this 3-letter sequence, instead of UGC, it was UGG. And so instead of alanine getting brought in, that ALA, it brings in glycine. This is called a missense mutation. A missense means 1 amino acid changes. Everything else stays the same. Arginine would still get brought in. The next 3 letters would still bring in the correct amino acid. So you could have 1,000 amino acids, 2,000 amino acids or more, and out of every single amino acid, all 3,000 or whatever, only 1 is different. If you're like, well, that's not so bad, depending which one it is. Sickle cell anemia is a missense mutation. One letter changed, one amino acid changed, and that changes the whole shape of the red blood cells. So depending on what changes, it can still be bad. Other types of changes for point mutations. So again, there's your original DNA, original messenger RNA, original amino acids. Well, now I'm going to change, after, right in the middle, there was an A. I'm going to make that a T. And everything still goes the same. Phenylalanine gets red, that UUU. But instead of the 3 letters UAU for tyrosine, it's now a UAA, which is one of the 3 stop codons. That's called a nonsense mutation. Nonsense means a stop codon gets put in when it shouldn't have. That can be detrimental because that means that nothing else gets made. There's no way that protein is going to do anything like it was supposed to because it's missing most of it. A frame shift mutation means you're either going to add one base or remove a base. We're not just going to switch the letter. We're just going to throw an extra base in or remove a base. So on here, we had 4 A's at the very beginning in the original DNA sequence, but now we're adding a T where there was no T before. So that 4th A is still there, but we've added a T in here. The first 3 letters are unaffected. That's still all the same. But now the rest of the sequence, it gets shifted over. So instead of the next 3 letters being ATA, now it's TAT and then AUA. It shifts everything over, which means pretty much after that insertion, every other amino acid brought in is going to be incorrect. This is because we're reading it 3 letters at a time, but now you shifted them over. So the first letter is not the same, the second letter is not the same, the third letter is not the same because we shifted them over 1. And that same thing happens with the frame shift deletion. I removed that T, that 4th letter up there, 5th letter. I just removed it. It shifts everything over. It's called a frame shift for a reason, which means everything after that mutation is going to be an incorrect amino acid that gets brought in. So although one change in your DNA sequence, 1 letter, 1 base, depending on which it is, can still be extremely deadly. Now, how do mutations occur? So sometimes mutations can occur spontaneous. It just happens somewhere in DNA replication, a base just gets brought in incorrect. Things that can encourage or increase the chance of a mutation happening is called a mutagen. And then it's got that root word or gen for generating, which just means it's generating mutations. Different types of radiation, there are different types of radiation, can change DNA sequences. UV light is a type of radiation, which is why if you're out in the sun, it can change the DNA in your skin cells. Luckily, our body can go in there and be like, oh, that's wrong. It can replace the incorrect DNA and replace it again, hopefully. There are chemicals that can cause mutations. Some chemicals will just change mutation sequences altogether, so they just alter the nucleotide. And there are some chemicals that have nucleotide lookalikes. So instead of bringing in the correct adenine, it brings in something that looks like it. So now you have a mutation, because it's not the actual base that gets brought in. If you see something known as a mutant or something known as a wild type means the normal, original DNA sequence. Which to me, I always think it sounds confusing. You're like, wild type? Well, that just means like it should be crazy. But it's not. It's the normal, like out in nature, out in the wild, the normal DNA sequence. A mutant means a changed DNA sequence. It has a mutation, so it's considered a mutant. So mutations don't occur all that often, but anything, whether it's a mutagen, whether it's some type of radiation or some type of chemical, increases the mutation rate. Now again, if you have mutations, generally changes in DNA is always bad. UV radiation on your skin is not great. Again, we have immune system cells. We have cells that can check DNA for mutations and either just remove those cells or kill them. But anything that increases mutations increases cancer risks. Because again, you change DNA. Now, my last little part of the PowerPoint are how can DNA get from one cell to another? So we've made DNA, we've translated it, we've transcribed it, flip those around. Mutations can happen, but how can we get DNA from one cell to another cell? So genetic recombination is when we're just exchanging information inside of one cell, which happens in more of eukaryotic organisms. When you have homologous sequences, this is information that shares in similar genes. Example, you and your cells, pink. You've got 23 pairs of chromosomes. Where'd your chromosomes come from? Mom and dad. So you have 23 pairs of chromosomes. One came from mom, one came from dad. They get paired up for a reason, because on the chromosomes, 23 pairs, this pair codes for similar information. This pair has genes that codes for your eye color, your hair color, everything else. But it can happen when you have two chromosomes hanging out by each other that shares in similar information. They can switch information. Like some of the information from hair color can go on to that gene, and some of that, I've already set my blue down somewhere, can go on to that one. And I'm like, and you can change up DNA sequences. It's why you guys don't all look like your siblings. Otherwise, really, well, you all got a gene from mom and you all got a gene from dad. Why don't you guys all look identical? Information gets flipped around. That's genetic recombination. Bacteria don't do that, but eukaryotic cells do. Vertical gene transfer, when you're thinking vertical, we're thinking top down, means that information goes from parent to offspring. This happens in eukaryotic organisms. Bacteria don't have babies. We don't have recombination of genes. They're just replicated and, they grow and divide. But vertical gene transfer is you have that information in those genes and you pass it on to your offspring, top down. Vertical gene transfer means it goes across. There's no offspring. It's one adult bacteria gives genes to another adult bacteria. So instead of top down, it's vertical. Horizontal, sorry. And when we talk about bacteria, because they don't have babies, this is how information gets passed on from one adult bacteria to another adult bacteria. So DNA can go from one bacteria to another bacteria. Now, because of prokaryotic organisms, it is the more common method of genetic exchange because they don't have infants. But there are 4 kinds of horizontal gene transfers that can happen. For my next 4 slides, and then we're done. Transformation, it's still going from adult bacteria to another. If you have 2 bacteria next to each other, here's my 2 bacteria. Normally, DNA, here's my DNA. Well, I'll make it circular. DNA cannot just leave the cell membrane, cell wall, transfer through another cell membrane, cell wall, and go into another cell. It's a lot of stuff that DNA would have to get through. Most bacteria can't do that. Some can, though. Some bacteria are actually almost leaky, and DNA can leave the cell and go into another cell. That's what transformation is bacteria DNA leaves through the cell and goes into another cell. And that's rare, but it happens. This is showing an example. So something happens to the cell wall, the cell membrane, DNA can leave, DNA can go in. How they figured this out is here's a bacteria that makes a capsule. Is a capsule good for the bacteria? Yes, so is that good for us? No, so the mouse dies. Bacteria that we killed If you take that same bacteria and you killed it, they heat treated it, they killed it, and they put it into the mouse, mouse lives. But there's another strain of this bacteria that doesn't make a capsule. If the bacteria doesn't make a capsule, it can't hide from the immune system, it won't survive, and the mouse will live. But what we did was we took some of these different kinds of bacteria of whether they killed the mouse or not, and we put them together to see if that information, if DNA for making a capsule can go into another cell. So we took a living strain that doesn't have a capsule, mouse lives. We mixed it with dead bacteria, so they didn't kill the particular capsule strain that didn't kill the mouse. We put them together, let them hang out for a little while, and we put it into the mouse. Mouse lives here, mix them together, mouse died. What happened is you still have living bacteria. Even though this doesn't kill the mouse, you mix it with these dead bacteria. And this dead bacteria, even though the bacteria is dead, there's still DNA present. And this DNA coded for how to make a capsule. And the DNA, the gene that codes for making a capsule, went into, which is rare, but it went into the living cells. And so now you have bacteria into the living cells. You have bacteria that weren't able to make capsules before, now making capsules. So it's bacteria can leave the cell and go into another cell. Rare. Transduction looks like a lot of steps, is using viruses to move DNA. When a virus hijacks a cell, the DNA from that virus goes into that bacteria cell. That bacteria is now a virus-producing factory, but sometimes instead of just viral DNA going into the new viruses that were made, bacteria DNA ends up in those viruses. And so when that bacteria dies, viruses leave, they go everywhere. Well, maybe that virus had some bacterial DNA. And when that virus goes and attacks another bacteria, it's injecting new bacterial DNA into it. All that often in the wild, but we know it can do this, and we play with DNA in labs. Bacterial conjugation is what we saw in the lab in that Hunting the Nightmare Bacteria video. Bacteria use a conjugation pili to connect two bacteria together, and information from plasmids can leave one cell to the other. And that's what was happening in that NDM1 gene. It was a gene on a plasmid that was leaving from one cell to another. And then the last is moving DNA, not just from one cell to another, say using plasmids, but moving DNA from one place to another, even in the cell. So we can take this DNA sequence, some gene that codes for something, and we can move it to another segment of that DNA. Sometimes we can even take that segment, duplicate it, and move it to other segments. The problem with transposons is when you're moving DNA from one spot to another, it can cause frame shift mutations in other genes. Usually the sequences that can get removed, copied, and moved elsewhere and then insert elsewhere are palindromic sequences. You guys remember what what is a palindrome? I give an example. Yeah, so if I had like A, U, 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 A, like it's read the same way forwards and backwards, which usually allows it to flip around and then insert elsewhere. But it can cause lots of frame shift mutations when it happens, but it still happens. We can still move DNA around.